Barely 6 weeks after the first recorded death from the new coronavirus in Wuhan, researchers in Australia will begin testing a potential vaccine on animals. A factory in Melbourne has started producing a test dose of a potential coronavirus vaccine, which scientists will begin testing on animals. The test vaccine was developed in just 6 weeks by University of Queensland researchers using world-first molecular clamp technology invented in Australia. Historically, making a vaccine has been a lengthy process, arduous, frustrating, and often unsuccessful. It takes about 10 years to bring a vaccine all the way from research through to approval, and that's if the vaccine works. Many more end up as failed experiments. The blueprint for the test vaccine was sent to the manufacturing lab in Clayton, and then the vaccine production began. The University of Queensland team say they have not perfected their design yet, and more tweaking will be done over the next period. The batch being brewed at Clayton is merely the first test dose, a process done in part to trial the factory's system. Nevertheless, it shows the incredible progress made by the researchers. Less than 2 months after the first recorded coronavirus death, a test vaccine is in production. After emerging in Wuhan in December, COVID-19 has spread rapidly around the world. In this video you can't see the virus, it's too small. The cells sit quietly in a bath. Slowly at first, but then more and more quickly, they start turning black until the entire dish is filled with death. The University of Queensland team began work on their vaccine on January 11th, when the first coronavirus genome, its DNA blueprint, was uploaded to the internet by Chinese scientists. "We did not need the virus itself, all we needed was the sequence," said Professor Paul Young. They quickly identified a section of the virus, known as spike protein, they would need to target. To do this, they are using molecular clamp technology, an invention developed at the Queensland University 7 years ago. The clamps are designed to hold the complex, changing the shape of the spike protein so the human immune system can get a good look at it and learn to kill it. Our immune system works by recognizing that protein and making antibodies to kill it. Specifically, you want your antibodies to recognize and kill the coiled shape of the spike proteins, killing the coronavirus before it gets a chance to penetrate your cells. If you want to give someone immunity without infecting them, the obvious thing to do is cut off a spike protein and stick it in a vaccine. But it's not that simple. Put a coiled spike protein, unconnected to the surface of the virus, in a vaccine and inject it, and the spike proteins will automatically uncoil inside the body. Rather than making antibodies to fight the coiled protein on the surface of COVID-19, the body will make antibodies that match the shape of the uncoiled protein. If full coronavirus comes along, those antibodies won't kill it. It is a fascinating and quite amazing technology, said a molecular biologist at the University of Auckland who is not involved in the project. This is done in huge fermenters filled with cells. The facility houses a 200-liter fermenting vat that will be filled with a soup of Chinese hamster ovary cells, the best cells for the task, in a nutritious goo. The cells will absorb the DNA and copy the instructions it contains, manufacturing liters and liters of the clamped protein. That will then be extracted, purified, and placed in a vaccine. This is the first candidate of possibly many, said research team leader at the manufacturing division. The coronavirus is also mutating, much as the flu mutates to escape our immune system. By generating a whole range of different antibodies, you should be able to get a broad immune response that can still handle a mutated coronavirus. The CEPI coalition has 3 vaccines for coronavirus in development around the world, with the hope that at least one works. 2 of them being developed by pharmaceutical companies working with CEPI, Inovio and Moderna, are of a type known as DNA vaccines. The other, being developed in Australia, is the molecular clamp vaccine. Other companies in China and Russia are independently working on their own vaccines outside of the coalition. "We feel we could be doing testing in animals by mid-March," says the Director of Health and Biosecurity of the facility. The technology works, on paper. Scientists are very excited. Several told us the University of Queensland vaccine had at least a 50-50 chance of succeeding. But the road to scientific discovery is paved with failure. 2/3 of new drugs that work in animals simply don't work in humans. So the scientists at CEPI estimate they would need to test as many as 21 different vaccines to have a good chance of one working. Here's the hope though, they only need to succeed once. Once the team's vaccine is finished, it will be tested on animals, first for safety and then for effectiveness.